Hello, everyone. I'm Mourya Nunna, and this is my thesis defense for the efficacy of stem cell therapies on ALS. And again, I'm Mourya Nunna. I'm a senior at Portola High School. And so the introduction is based on my research question: how can stem cells be used to reverse damage in neurons caused by ALS? So a quick introduction on ALS. It's a neurodegenerative disease that targets the motor neurons in the brain and spinal cords. So motor neurons are basically the neurons that are connected to all the muscles and facilitate movement in the body. And during this research, I was investigating the types of stem cells and their efficacies. And the goal of the study was to find how neural stem cell transplantation would decrease the progression of the disease. So I was focusing on how it could slow how fast the disease goes throughout the body and slowing the damage it does. And current medication for ALS is limited. There's only a handful, less than 10 approved drugs by the FDA to be used for ALS, and all of them focus on slowing the progression of the disease. None of them are built for actually stopping and curing a person with the disease. And the literature review was I analyzed many studies focusing on current or past trials in animal models and humans. And throughout the paper, I talked about the specific types of stem cells and ALS. I identified the rates of success in patients with each type of ALS. So there are specific mutations, such as the SOD1 gene, or there are cases where it's just sporadic. People randomly get it with no underlying cause, no genetic history. Nobody in their family has it. They just randomly get it through mutations or some other factors that still haven't been discovered. So on the bottom left is a picture of the aforementioned SOD1 protein that the gene is responsible for manufacturing. And I analyzed research that was based on intrathecal injections of neural stem cells that are transplanted into the intrathecal region of the spine, indicated on the picture on the bottom right-hand corner. And so for the methodology and results, just like I said, I analyzed the results from transplanted stem cells in human and animal models, and results show that stem cells are able to integrate with the host environment with minimal side effects. So when transplanted into human or animal models, the stem cells mimic the actual neurons in the human body and are able to connect with them without having any side effects. There have also been known to have close interactions with the normal neurons, releasing neurotrophic factors to protect the already damaged neurons and slow their rate of decay. And the results were analyzed from multiple studies, indicate that transplantation prolongs the life of the host and returns varying degree of muscle control back. Important thing to note here is that it does not cure any of the hosts from ALS. It simply prolongs their lifespan and prevents the accelerated decay of the motor neurons. And the conclusion, again, the research question is how can stem cells be used to reverse damage in neurons caused by ALS? And the research was done through analysis of clinical trials of animal and human models to support my thesis, and findings where stem cell transplantation is a viable solution to reversing the damage caused by ALS. Still has a lot of potential to be developed. It is not perfect right now, and there are limited trials that are approved by the government due to the nature of the disease. And stem cells, although they're not new, stem cells still have a lot to be discovered. So clinical trials in humans are not widespread across the country. And it's also important to note how stem cell therapies are not exactly a cure to the disease. Rather, they reverse the damage done by it. So until a proper cure can be discovered for the disease, using stem cell therapy to reverse the progression and return some muscle control back to people so they can live longer is the focus of my research. And research experience, I'd like to thank Dr. Berkey and Professor Moshe for their guidance. All the lessons throughout the 8 weeks were really interesting. They taught me a lot about how to conduct research and how to write the paper, especially Dr. Berkey's lessons and diving into the deeper and more complex topics while making it simple to understand were really helpful. So thank you to both of them for guiding me through this research. Thank you.
Excellent. You want to turn off your presentation? Dr. Varki, question for him.
Mourya, good job, on this ALS. Just a question: when you looked into ALS, so one is like SOD1 gene that you might have looked into. Did you also find some other proteins involved in ALS apart from SOD1, which they might be important for, which you might consider as important for treatment purpose?
Yeah, I mostly looked into the SOD1 gene because normally when the protein created from the SOD1 is released, it mainly is supposed to help the neurons, but in this case, the misfolding causes them to kind of cause negative effects against the neurons. So I mostly focused it on that because in animal models, it was mostly the SOD1, so I didn't really look into other types of proteins in ALS. So it was mostly focused on the protein released by the SOD1 gene.
Okay, good presentation, Mourya, and I also wish you best wishes for your future. Let me know. It's just for all the students who are in presentation today. Even after today's presentation, just to let you know, if you have any questions, if you need any clarity, you can always contact me through Discord. Your channel is always active there. Good job.
Thank you.
Thank you, Mourya. Thank you. Now that your research presentation is done, your essays need to be submitted. I'm just kidding. I'm after your life. No, you're doing good.